Hello and welcome to Virtual Investor Conferences. My name is Eric May, and on behalf of OTC Markets, as well as our co-host, Zacks Small Cap Research, we're very pleased you have joined us for our next live presentation from NeuroBo Pharmaceuticals, Inc. Please note, you can submit questions in the box to the left of the slides. You can also view the company's availability for one-on-one meetings through the Schedule Meetings tab found on the conference platform. At this point, I am very pleased to welcome the next presenters, Bob Homolka, SVP of Clinical Operations, and H.H. Kim, CEO and President of NeuroBo Pharmaceuticals, which trades on the NASDAQ under the symbol NRBO. Welcome, Bob and HH.
Thank you. Hello, everyone. Hyung-Han H.H. Kim, CEO of NeuroBo Pharmaceuticals, listed in NASDAQ with the ticker of NRBO. We have Bob Homolka here as well. Let me start with the introduction. To start with, we would like to highlight the strong leadership team that we have inside of NeuroBo. There have been some turmoil during the past 2 to 3 years, but now we have a solid team. I myself, as the Chief Executive Officer, we have Mi-kyung Kim, the CSO. She is the person in Dong-A Research Center who made these 2 assets in the company from scratch in-house. Romot Homolka, SVP of Clinical Operations, with amazing amount of experience in clinical trials. He's leading the charge at the moment. We have Dr. Stephen Harrison as Consulting Chief Medical Officer. I'm pretty sure everyone knows of Dr. Harrison, who's interested in the NASH field. He's the leader in the NASH field. And we have Marshall Woodworth, Acting Chief Financial Officer. And we have Sungjin Kim as Director of Corporate Strategy. Now, I'd like to focus on the compelling investment opportunity that we propose. We have targeting MASH and obesity with a pipeline of next generation therapeutics. We're aiming to increase shareholder value through multiple near-term value-creating milestones. DA1241, we have an open IND for treatment of MASH and type 2 diabetes, actively recruiting into phase 2A for 1241 in subjects with presumed MASH. We have completed SAD and MAD studies in healthy volunteers and subjects with type 2 diabetes as phase 1 study in the US. That's the reason why we have 2 open INDs for MASH and type 2 diabetes. For the second asset, DA1726, submission of IND for obesity is targeted to be late December 2023. We have a strategic partner and major shareholder, Dong-A ST, as our biggest ally and partner. They provided with these 2 assets, DA1241 and 1726, in a licensing deal, and they did 50% of the financing we completed during last year. We are well capitalized with $25.8 million in cash as of end of third quarter 2023. We have cash runway going into the fourth quarter of next year. Now, with other legacy assets such as the botanical products, which is NBO1 and 2, and we have the COVID asset, which is ANA001, we are actively exploring strategic opportunities so that if there is any chance to have these assets going forward, we're open to hearing it. Now, for the 2 assets that we are actively developing, we have DA1241, indication in MASH. It is a GPR119 agonist. Finished with the preclinical. We finished the phase 1 in the US, and now we're actively recruiting in phase 2A. DA1726, an obesity drug, GLP-1 receptor, glucagon receptor dual agonist, finished the preclinical, and we're targeting to submit the IND within this year. Now, multiple near-term milestones. Investment into the current DA1241 phase 2A and planned DA1726 phase 1 have the potential for significant returns in the event of clinical and regulatory success. For next year, DA1241, we're looking at last patient, last visit during the third quarter. And we are hoping to have the top-line results within the fourth quarter of next year. Going into 2025 with DA1241, we'll be having meetings with FDA to analyze the top-line data, and we are hoping to go into a phase 2B protocol submission in the later half of 2025. Now, for DA1726, as mentioned, we are targeting to submit the IND within this year, which is this month. Then we are hoping to have an approval for the IND in the first quarter of 2024, and hope to have the first patient in for phase 1 part 1 during the first half of next year, and have first patient in for part 2 of the phase 1 in the second half of next year. With all that activities, we are hoping to see the part 1 of phase 1, last patient, last visit during the first half, and hope to have the top-line results within the first half of next year, 2025. And during the second half of 2025, last patient visit for part 2, and top-line results during the second half of 2025. Now, this is a little bit different than what we have presented in the past. The reason for a comprehensive phase 1 study that goes beyond 2024 is because of the known side effects of GLP-1 receptors. And with the FDA having been cautious about starting a GLP-1 receptor trials in humans for the first time, we are making sure that we control the side effects, make sure that we find the right dosing. Now, starting with DA1241, it is orally available potential first-in-class GPR119 agonist for treatment of MASH. Starting with competitive differentiation, everyone knows in the MASH field that resmetirom, Madrigal's asset, successfully went through with phase 3, and I believe they will be starting some activities and marketing next year. For Madrigal's asset, efficacy in human was MASH resolution with more than a 2-point reduction in MASH activity scores. And for our asset, what we're trying to look at during our phase 2A is effectiveness in treating or modifying the progression of MASH, MAFLD activity scores, and biomarkers. We believe that DA1241, compared to the Madrigal asset, has a unique mechanism of action working on inflammation associated with MASH, and can be used as a monotherapy or in combination with other therapies, since synergistic effects can be seen when co-administered with a DPP4 or a GLP-1 inhibitor. Looking at DA1241 effect on the pathogenesis in MASH as a monotherapy, GPR119 activation that will relate to macrophages activation, monocyte recruitment, macrophage differentiation will result in reduction in hepatic and systemic inflammation. Hepatic stellate cells will stop stellate cell activation, which will reduce hepatic fibrogenesis. And the hepatocytes and intestinal L cells activation will stop de novo lipogenesis and dietary fat absorption, resulting in reduced hepatic steatosis. So on the 3 stages of MASH, steatosis, steatohepatitis, and fibrosis, We will be looking at different biomarkers to see the effects, since we're not doing biopsy for the phase 2A, and I will show how the results come out. Now, with a combination therapy, GPR119 and glucose control, we did do preclinical studies with DPP4, sitagliptin. That's the reason why we have part 2 of phase 2A with combination therapy. With combination, we do see effectively decreasing hepatic inflammation, reduced systemic inflammation and fibrosis biomarkers, and reduced hepatic lipid and collagen deposition in liver of MASH mice. So GPR119 will activate GLP-1s in natural forms, while a combination with DPP4 inhibitor will interfere with inactivation, meaning that prolonged GLP-1 activation could be heightened. So we have a combination with 2 means more natural GLP-1s, while DPP4 continues or tries to continue the activation. So having those in it, we did look at more than 20,000 gene expressions. When you look at the left corner, you can see that in inflammation signals, there is a very good heat map there, and especially with combination with DPP4. When you look at stellate cell activation related genes, it is quite impressive, although not as much as inflammation, but we can still see improvement in that area as well. And stellate cell, that means, is the later stage of MASH where there is no cure at the moment. Now, for DA1241, with looking at the combination, we did compare it with elafibranor, which was being developed with more focus on inflammation. You can see that inflammation scores can have 3 positive areas. You can see that as a standalone, DA1241 has good effects compared to elafibranor, but with the combination, it does show a much better results. A little more detail on the combo side. Due to the time restraints, I'll go right into just one factor here. As I mentioned, activation of natural GLP-1s and DPP4's activation to prolong the activities of GLP-1. You can see on the left side that as a combination, active GLP-1 levels are much higher than DPP4, which we are hoping to see results in a variety of different areas. I'll hand it over to Bob.
Yeah, thank you, HH. As HH mentioned, we are trying to distinguish our 1241 from our competitors in 3 ways. One is a unique mechanism of action. 2 is it can be used as a monotherapy or as in combination. And third is to really look at inflammation markers. The phase 2A study actually is looking at all those, evaluating all those areas. This is a part 2 study, a 2-part study in NASH patients. The first part is 1241 at 50 and 100 milligrams versus placebo. The second part is the combination arm, where it's 1241 and sitagliptin against placebo. The study is expected to randomize approximately 90 patients in the United States, and we are targeting 25 centers as well. We hope to have the last patient last visit in the second half of in June of 2024. So back to you, HH.
advantages in MASH. Just to wrap it up, GPR119 targets inhibit steatosis, inflammation, and fibrosis. And it is an advanced GPR119 agonist. And as you've seen, it has a great potential with a combination with DPP4 inhibitor. Now, going into DA1726, novel GLP-1 receptor, glucagon receptor dual agonist for treatment of obesity. We'll start with the competitive differentiation. On the left side, you can see that survodutide, mazdutide, these are similar GLP-1 receptor, glucagon receptor assets. On the right side, you can see semaglutide, GLP-1 receptor, and tirzepatide with a triple agonist. When you look at survodutide developed by Boehringer Ingelheim, you can see the efficacy in humans and the safety in humans as well. And when you look at the safety side, nausea, vomiting, diarrhea, you can look at mazdutide, Eli Lilly developing it, similar side effects. And when you look at semaglutide, tirzepatide, you also see similar side effects as well, nausea, diarrhea. And when you look at semaglutide with GLP-1 receptor alone, nausea, diarrhea, vomiting, constipation. So what we're trying to see during our phase 1 and in our clinical trials is that our DA1726, although has similar GLP-1 receptor and glucagon receptor, we are more balanced in those two receptors, and that we can show, and we're hoping to show, that the side effects can be minimized compared to other competing assets, but with similar weight loss. Then it will result into it will be better tolerated than semaglutide. Going into DA1726, novel oxytocin analog functioning as a GLP-1 receptor, glucagon receptor dual agonist for the treatment of obesity. Now, GLP-1 receptor will reduce food intake, while glucagon receptor will increase the energy expenditure, potentially resulting in superior body weight loss. Going right into the comparison with semaglutide GLP-1 receptor, when you look at the left side, you can see that cumulative food intake in HF obese mice. Semaglutide was 72.1%, meaning almost 30% of food intake was reduced, while 1726 high dose was 80.2%. That's about 8% difference. Now, when you look at the middle graph, body weight loss, 1726 higher dose was about 8% more weight loss compared to semaglutide, which was about 19%, meaning the mice with 1726 ate about 8 to 10% more, but lost about 8 to 10% more than semaglutide, which we would like to show in our clinical trials that more food intake means less side effects, and the glucagon receptor has effect on higher energy expenditure, resulting in more weight loss. Now, with a tirzepatide comparison, you can see in the middle, cumulative food intake, tirzepatide, 58%, while high dose of 1726 was above 75%, meaning there's about 20% difference in food intake. But when you look at the left side, body weight loss, it's the same, meaning similar to semaglutide comparison data, the mice on 1726 ate 20% more food, but lost similar weight loss that has been shown with tirzepatide. Bob?
Thank you, HH. As HH mentioned earlier on in the presentation, this is a first-in-human study, so we are being cautious especially with this class of drugs, so we understand the effects as well as the side effects. So the first-in-human study is a classical SAD and MAD study where the first part is a single ascending dose. The second part is a multiple ascending dose study. We are testing this in subjects with obesity, but are generally healthy, and this is dependent on the number of cohorts we go. But we're expecting about 100 of subjects to participate in both parts. It is located. There will be a single center in the United States, and we look forward to starting that study up. It's
Thank you, Bob.
it's expected to start in the first half of 2024, as HH mentioned above. Over to you, HH.
Thank you, Bob. Just to reiterate what we would like to present to the highlight of 1726 is that we saw more food intake by the obese mice models while losing a similar weight loss. We believe that that relates to reduced side effects, and hopefully that because of the more energy expenditures, we would like to see the results of the clinical trials translating over to more food intake. If it's more balanced, healthier diet at the end, because the food intake wasn't reduced as significantly compared to other assets, that the rebound should be lower. Plus, the muscle loss shouldn't be as severe as other assets out there. So that's the key that we would like to convey to potential investors. Now, going into financials and capitalization, as noted in the front, financial overview, as of September 30th, end of third quarter, in cash, we have $25.8 million, and we don't have any debt. Now, compelling investment opportunity, DA1241, it is focused in inflammation compared to the other assets out there. And with a combination therapy, we believe that we can show it has great potential in the MASH field. And DPP4 is a candidate for combination therapy as we are going through it, phase 2A, part 2. But there are potential out there that we can have a combination where we can attack the phase 3 and 4 of MASH. So there is great things to be done, but we'll take step-by-step approach, make sure that we finish the phase 2A with great results, and we'll look what we can do after that. On 1726, next year we're starting phase 1. The preclinical data on obese mice models, it does show a great competitive edge compared to other assets out there, especially looking at the food intake wise and on the body weight loss. That has a very big potential to be moving forward with our clinical trials and making sure that we show our hypothesis right. Thank you very much, and we'll go through with the questions. I'll start with, do you have any research partners, and are you attracting big pharma interest in TA1726 development? Now, as a listed company, we won't be able to provide you with information that's not being disclosed. But the biggest research partner would be Dongha ST Research Center that has developed these assets from scratch. They have the capabilities of preclinical, in vivo, in vitro data to be generated. And we are working closely with Gubra, which has great mouse models to be utilized. And as similar to other obesity-related companies out there, I'm pretty sure everyone's talking to each other. What types of strategic opportunities are you exploring? I guess that will be related to the assets, I believe. Asset-wise, 1726 is still too early. We would like to see in-human data before we'll be able to discuss in detail with anyone out there. DA 1241, we are seeking what kind of combination can we get to really tackle the phase 4 of MASH? Fibrosis is a very hard area to be tackling, and we do know inflammation has a great effect in going into fibrosis, so that we know we can control somewhat of the earlier stage to the middle, but patients who went into fibrosis, we don't really have an asset out there really can tackle it. So our focus will be looking into a combination therapy that can boost the inflammation while controlling the fat side of MASH. The market for DA 1241, what is the total addressable market? I believe it's the MASH field. It's a huge indication. It's in the billion-dollar range of market that finally Madrigal has been able to, or it will be tapping into. But I do believe that one asset alone can be the cure-all for the MASH disease. Now, it used to be NASH, but the scientists are calling it MASH because it's more metabolic disorder. And in that angle, 1241 has a great chance to penetrate this market. And I believe phase 2A data will hopefully show that. The last question, who is Donga ST? Donga ST is part of a group called Donga Social Group in Korea. The group of companies were established in 1932. Established the first pharmaceutical R&D center in South Korea back in the 1970s. Donga ST itself is focused on prescription drug development, manufacturing, research, sales, and marketing as well. And they are the big strategic partner for our assets, plus with the financing as well. Now, having a great partner like that, it gives us a good scientific and research center capabilities that we don't have coming in. We have shared services agreement where they provide us with all the help that we need from the research center, and that really helps us as a building block for this company. I think one last question. What are the advantages of your drug compared to other being developed by other companies? I believe 1241, I've told you, we're more focused on the inflammation side. 1726, the comparison in the preclinical data is that the food intake did not decrease as much as other similar analogs and GLP-1. So that will be focusing on that and trying to translate that into human data and show you our results. Okay, I think that will be for the day for us from NeuroBo. It was great meeting everyone, and if you have any other questions, please feel free to relay it over to us. We're more than happy to meet investors who are interested or anyone else who is interested in our company. And I hope everyone has a great day. Thank you very much.